Good morning. Why so less? Only 15 people for the test? Well, the test would not be that difficult, but it would be very interesting. Okay, so let's start. Hmm, What? So all good? Right? You can hear me properly? Next marathon is on 29th of January. Areva, wish you a very happy birthday from all of us, Siddhant. Okay, so as usual, right? This time there would be 55 questions. There would be 55 questions. And yes, please do not write answers in chat box, right? So, and wish you all the best. The most important thing is this time, not more, but at least say 4 or 5 questions would be where I'll be giving you almost 1 full minute, or if you want, you can have even more time, right? 1 to 2 minutes that can be given, because it would be actually a descriptive question in which you have to actually write at least 3 4 sentences, right? It won't be like one word. So this is one. There would be few questions where you have to analyze. Analysis is necessary. So over here also you'll be given more time. The questions which are absolutely straight or either one word, we'll go through those questions bit fast. Okay? So but do remember this, that do not write any answer into our chat box, because that's very important. So wish you all the best, and we start, because 55 questions, so it will take about an hour or so, and then we'll be discussing, right, each and every question. No worries. All you have to do is in your book, you just write 1 2 3 4 that way, and you have to just write the answer in your book. Okay? You have to just write answer in your book. You don't have to write anything into the chat box. That is first. And second, when we'll be discussing, right? When we'll be discussing, it is like you'll be self-evaluating your answers. No one will be there to check whether you have answered it or correct, right? It is like honesty. I trust you. So if you have answered very properly, precisely, without any spelling mistake, right? Do remember, without any spelling mistake, without spelling mistake. This is very important. So if you have done that, so then you give full mark to yourself. And if there is any error, so then mark it, right? And do write the correct answer also. So it will be helpful. Next time you won't be making that mistake, correct? So that's the thing. And bus all the way, you have to reach up to the final question, 55. In case if you come across a question which you find that you don't know the answer, right, you can keep it blank, because while discussing it, then you can take care of that question. Okay? The idea is you are competing against yourself. There is no one to fight with. You are competing against yourself, and this is like a very simple assessment that how well you have understood the topic, right? I got 2 3 emails, and well, we'll talk about it later on. And but just one sentence I would say, and that is we can't control how others are teaching, right? So why not to focus just you and me, right? In case if you find any problem, just let me know and I'll explain that thing again. But well, I can't change other teachers, right? That is not possible. So please don't, means I totally understand, but say because this is a public platform, we can't speak about such things. But yes, when the only PowerPoints are used, surely it's a problem, right? But we'll manage, right? At least say when you have put so much of faith, and I'll try to give my best. Okay, thank you. Let's start. And here is the first question. So all set, everyone ready? Right? If you are not written from 1 to 55, no worries. Sat you can write it. Right? So done. Let's start. Okay. Here is the first question. And I start the timer also. First question. What is microbiota? What is microbiota? And I know this is slightly descriptive because you have to write down those keywords. So take your time. Okay. Next question. Question number 2. ICF compartment consists of fluid between the cells. True or false? It is just true or false. That's it. No description needed. ICF compartment, intracellular fluid compartment, consists of fluid between the cells. On to the next one. ECF contains chloride and CHO3 minus. True or false? ECF contains Cl minus and CHO3 minus. True or false? Then the next one. In control mechanism, this is question number 4. In control mechanism, which cycle is dangerous and may lead to death? In case if I am going fast, just let me know, right? You should be able to write comfortably, right? We are not at all in a hurry. So in case if I am going fast, immediately let me know. I'll just slow down. Question number 4 is, in control mechanism, which cycle is dangerous and may lead to death? On to the question number 5. For question number 5, you'll be given 1 full minute because you have to give an example of positive feedback mechanism which is useful for the physiological process. You have to write down an example of positive feedback mechanism and which is good, which is useful for the physiological process. 1 full minute. 15 seconds to go. Okay, on to the question number 6. Who is responsible for the grainy appearance of rough endoplasmic reticulum? Next question, that is question number 7. Which part of the cell membrane is lipophilic? Which part of the cell membrane is lipophilic? On to the question number 8. Sphingolipids, they are present in which type of cells? Sphingolipids are present in which type of cells? Question number 9. Okay, give any two functions of glycocalyx. So you can have about half a minute or even 45 seconds. Give any two functions of glycocalyx. And yes, if you know more, you can definitely write more than two, right? But minimum two functions of glycocalyx. Still, you have got another 25 seconds. Right. Question number 10. Quite easy. What is synthesized by rough endoplasmic reticulum? What is synthesized by rough endoplasmic reticulum? Quite a straightforward question, so we'll be moving a bit fast. On to the question number 11. Hydrolysis of lipids leads to formation of which substances? Hydrolysis of lipids, right, leads to formation of which substances? Remember, it is substances, so that means it is more than one. Next one. Again, what is the function of peroxisome? So you can, because there are few keywords to write in this, so you can have 45 seconds. Akram, don't write any question, any answer over here. Don't write any answer over here. Good morning, but don't write any answer over here. Right? You have to write answer in your book only, in your notebook only. You don't have to write. Now, no worries, that's fine. If you are joined now, so start from here. No problem. Start from here. Okay. We move on to the question number 13. Yes. This is mitochondria, right? So identify the structure which has been marked over here. Sure, I'll do that. Right? Identify the marked structure in mitochondria. On to the question number 14. In a cell, mitochondria are fixed in number, right? This is a statement. You agree with it or not? True or false? No need to give the reasons, right? Reasons we'll discuss when we'll discuss the question. Right now, just true or false. In a cell, mitochondria are fixed in number. True or false? Question number 15. Okay, here you can have about 30 seconds. What is desmin? Don't write, don't write answers over here. Mr. India, don't write answers over here. Write down in your book. Write down in your book, not over here. Okay. I have to. Ha. So on to the question number 16. Name the substance. That's okay, that's okay. No need to be sorry. You might not be knowing, that's why. So you just join. If you are joined now, continue from this question. Name the substance forming boundary of the nucleolus. What is name of that substance which forms boundary of the nucleolus? Question number 17. Okay, for this you can take about 45 seconds. Give 3 characteristics of pinocytosis. Again, if you know more, you can write more, but at least 3. 10 seconds have passed. Still you have got time. So give 3 characteristics of pinocytosis. Okay. On to the question number 18. What is the function of lysoferrin in lysosome? Lysosome has got some functions. In that there is lysoferrin. So what is that lysoferrin doing, or what is the function of lysoferrin? On to the question number 19. Okay, for this also you can take about half a minute or even 45 seconds. It has started. Time has started. By which processes, right? Remember, I've asked processes. SER, smooth endoplasmic reticulum enzymes, do the detoxification. There are few processes by which this is done. Which are those processes? Okay, move to question number 20. Yeah. What is synthesized by Golgi bodies which ER cannot? Right. Very easy. So just 15 seconds. Correct. Hmm. Question number 21. Okay, in this, right, you have to fill in the blank. You have to fill this space. Fill this space. There is a missing component over here. Right. So carbohydrate to glucose, lipids to fatty acids, proteins to amino acids. They are giving to this acetic acid. This acetic acid, right, joins here, leads to formation of this substance. This is X, and it leads to formation of acetyl-CoA. Who is this? Right? That is what you have to write. Okay. Question number 22. Name the cations present in ICF. 19th one? Sure. 19th. 19th is by which processes SCR, smooth endoplasmic reticulum enzymes do the detoxification. Right. Then it was what is synthesized by Golgi bodies, which ER cannot. That was 20th. And the 21st was you have to fill in this space which substance we are talking about. Right. On to the question number 20. Okay, 22 has already been shown to you. Right. Name the cations present in ICF. So you must have answered it. So let's move on to the next one. Right. Question number 23. Again a statement. ECF is double than ICF. Right? ECF is double than ICF. True or false? Just true or false. Next one. Name the substances which are used to estimate the total body water by dilution method. Right? Take your time. 30 seconds. Substances which are used to estimate the total body water by dilution method. Okay. On to the question number 25. Yeah. Again, it is true or false. Inulin cannot cross the cell membrane, while mannitol can cross the cell membrane. Is that true or it is false? You have to just say true or false. Bas. No reasons. Question number 26. You must have noticed, because if you know the answer instantly, you'll be able to answer in a second. Question number, you want question number 24? Yeah, here it is. Name the substances which are used to estimate the total body water by dilution method. Okay. Then this was that inulin cannot cross the cell membrane, while mannitol can cross the cell membrane. Yes or no? This one, 26. Okay. For this, take one full minute because this is conceptual. What is Gibbs-Donen effect? One full minute. And try to, if you don't write, to at least draw so that you know that you have got clean concepts about it. Okay. What is Gibbs-Donen effect? Full 1 minute. Still you have 15 seconds. Okay, you must have completed it. On to the question number 27. What do you mean by ubiquitous? What do you mean by ubiquitous? This U can be eliminated because if this is TOUS, then it is a British spelling. If you just write UBQITOS, then it is American spelling, but meaning is same. So nothing to worry about. What is the meaning of ubiquitous? On to the question number 28. Okay. Question number 28 is, name the substance which will use to estimate the intracellular fluid only. The substance which you use to estimate the intracellular fluid only. On to the question number 29. Calculate the osmolarity of a solution with following details. You got 1 liter of fluid, 2 moles of sodium chloride, 2 of calcium chloride, and 1 glucose. What would be the osmolarity? Okay. Question number 30. Right. Take your time. Take 1 full minute. Fill the below details when hypotonic solution is added. So what would happen to ECF volume, ICF volume, and what would happen to osmolarity of ECF and ICF? 1 full minute. If you have finished this, we move on to the next one. Okay. Yeah. Fill the below details when hypertonic solution is lost. Right? When hypertonic solution is lost. Okay. On to the question number 32. Which are the 3 conceptual criteria responsible for the substance to cross the cell membrane? Right? You have to write all the 3 criteria. These are the criteria which are responsible for the substance to cross the cell membrane, whether the substance can really cross the membrane or cannot cross the membrane. So these are the three conceptual criteria. Okay, you must have written 5 more seconds and we move on to the question number 33. In which part of the cell membrane phosphatidylserine is normally present? Right? Carefully read the sentence. What it is asking is, what has been asked is, in which part of the cell membrane phosphatidylserine is normally present? Okay. So no confusion. In a normal cell, when things are fine, where do you find phosphatidylserine? On to the question number 34. Saturated fatty acids, they increase the fluidity of cell membrane. True or false? Because it's an easy one, right? We move to the next one. Now the question number 35. Okay. In membrane of which cell is the presence of more lipids as compared to proteins? In membrane of which cell do you find more lipids as compared to proteins? You have to just tell the cell type, which cell. No explanation needed. Okay, question number 36. Right. Name the major surfactant. You have to just give the name. Major surfactant. So this was question number 36. On to question number 37. 37 is, which ratio is vital to check the fetal lung maturity? Fetal lung maturity. It would be very nice if you can write the full form. But even if you write short form, that's okay. What should be the ratio? Well, if you write, well and good. That's what I said. If you write the value, it is good. If you just write the name of the ratio, then also it is good. But if you write the value, very nice. Question 38. Which antibody is detected in VDRL test? Which specific antibody is detected in VDRL test? Venereal Diseases Research Laboratory test. VDRL test. Okay. Question number 39. Define receptors. You'll be given 1 full minute. You have to write down very properly what exactly a receptor is. We'll be focusing on some keywords in this. That is when you describe receptor, take care of those keywords. So take one full minute. You got still 45 seconds. So take your time. 15 seconds. Okay. On to the question number 40. GPR receptors are also known by two other names. You have to write those two names. One is GPCR receptors. Which are the other two names? Quite easy. Hmm. Question number 41. This question will never be asked in exam, in any of the exam of the world. What is bilog protein in GPCR? Because it is not given in any of the book, right? Because this is like a conversation between you and me. So, but yes, you know that what is bilog protein in GPCR, right? Or in other words, in GPCR, G stands for. I was so tempted to ask this question, क्या bilog protein. Hmm. Under question number 42. Hmm. What is the effect of inhibition of adenyl cyclase enzyme? What happens when adenyl cyclase enzyme is inhibited? Question number 43. Name the enzyme which is inhibited by drinking tea or coffee. Enzyme which is inhibited by drinking tea or coffee. Okay. Question number 44. Protein kinase A. It activates the enzyme to stimulate basic body metabolism. How is it done? Protein kinase A does what to enzymes so that they stimulate basic body metabolism? By which method protein kinase A activates the enzyme? Because it is that activated enzyme which will stimulate basic body function or the basic body metabolism. True. Question number 45. Easy one. What do you mean by mydriasis? What do you mean by mydriasis? Next one, question number 46. So that means last 10 questions. And I'm sure you must be doing good. So where do you find beta 2 adrenergic receptors in body? Where do you find beta 2 adrenergic receptors? Question number 47. 47 is, yes, take one full minute. Name 3 factors which are affecting diffusion. So write all 3 factors which are affecting diffusion. And do mention whether they are directly proportional or indirectly proportional. Right? Be clear in that. So mention factors which will increase, mention factors which will decrease that diffusion. Okay. Question number 48. All right, take your time. List the locations of GLUT type 2, glucose transporter type 2. You have to write all the locations. So try to write as many as possible. If you have remembered it properly, you'll be able to write all of them. Take your time. Okay, 10 seconds more and we move to the next question. So question number 49. Which syndrome results in mutation of GLUT2? Syndrome which is occurring due to mutation of GLUT2. Just the name of the syndrome, right? Name of the syndrome. Do take extra care for the spelling. Okay, nice. The next one, question number 50. Yes. List the location of glucose transporter type 5. Glucose transporter type 5. If you have not noticed, locations. Okay, locations. That's a hint. I not ask for location, it is locations. So do take care. Question number 51. Watch it. Right. Everything is mentioned over here. Everything is mentioned. You have to just say whether it is correct or not. Right. A cell membrane is shown. What are the type of processes? Everything whatsoever is needed is written. Just check out that everything is fine. If yes, say yes. If you find that something is not okay, so then say no. But don't answer here. Okay. Next question. Question number 50, 52. Right. Which part of nephron is responsible for the reabsorption of filtered glucose back in blood? Right. You have to just name part of the nephron which is responsible for reabsorption of filtered glucose. Because this is exam, I can't speak much, but focus on reabsorption of filtered glucose. Reabsorption of filtered glucose. All of you are geniuses, you know, and seriously, it would be a pleasure teaching all of you. Because if the concepts they are good and strong, right? Imagine how happily you'll be learning the medicine, the cardiology, the nephrology, the ECGs, ultrasonography, surgery, whatnot, everything. Okay, done. On to the question number 53. As such, this is the question, because but we have discussed, that's why I'm asking this, because usually this is the question for second MBBS, that your diabetic patient is on canagliflozin. Which common complication you'll watch for? Right, and nothing more is mentioned. So if you have studied, if you remember it properly, immediately it will strike that yes, this was the thing. So your diabetic patient and you have put him on canagliflozin instead of metformin. So now, which common complication you will watch for? The patient should not be having that. Right? Just name of the complication, no descriptions, nothing. Because if you write the name of the complication, that itself means you know the entire mechanism. Okay. Next question, question number 54. Uh-huh. Why cardiac arrhythmias can occur in gentleman's syndrome? Thanks, Sanskriti. Why cardiac arrhythmias can occur in gentleman's syndrome? There is a very specific reason why cardiac arrhythmias occur in this. Just try to recollect. Okay. On to now the last question. This last question is coming up 55, question number 55. Yes. So we are talking about Bartter syndrome, right? And we have got serum values and we have got the urine values. You have to just say which value will increase and which value will decrease. Bus. In Bartter syndrome, you don't have to write down the reasons. You have to just write what will increase in serum, that is calcium, whether it will increase or decrease, in urine, whether sodium will increase or decrease, magnesium, whether it will increase or decrease. That's what you have to mention. So fill up those spaces here, here, here. Anyone who has missed any of the question? If everything is fine, because this was the last question, question number 55, right? And it's done. So anything left out? 7, 8, and 16. No problems. No problem. Let me take that back fast. 7. Here is the 7. Which part of the cell membrane is lipophilic? Right? Which part of the cell membrane is lipophilic? Question number 8 was sphingolipids. They are present in which type of cells? Correct. Sphingolipids are present in which type of cells? And the question number 16 was, name the substance which is forming boundaries of the nucleolus. Right? Name the substance forming boundary of the nucleolus. Okay, all done. Anything left out, just let me know, and then we start discussing all the questions. It would be a sort of complete revision right from the beginning to end. It was intense, Deepak. But truly, I want all of you to prepare in such a way that any exam and you won't be having any trouble. So, okay, okay. So let's start, right? So what is microbiota, right? We remember that this was like microorganisms, what we were talking about, right? These are the microorganisms. And in GIT, we have got 400 to 1000 types, right? Yeah, exactly. Exactly. So that's what I really want you to prepare. You should be so powerful that even any of the exam should not be a problem. So 400 to 1000 types of microorganisms they are into our GIT, but some of them, right? Some of them they can cause disease. Yes, that's right, right? It can cause disease, right? But under normal circumstances, right? Under normal circumstances, they are in harmony, right? They are in harmony with others. So all is well. Correct. These are the keywords. Right? So they can cause disease. This was the main important thing. Microorganisms which can cause disease. On to the next one. ICF compartment. Yes, that's right. Live in harmony with human body, but they can cause disease too. Perfect. ICF compartment consists of fluid between the cells. If anyone has answered this question wrong, I have to remotely hit you. Ah. Because if anyone has written true, तो बस गया काम से, right? Because this is interstitial, the consist of fluid between the cells. Fluid between the cells is what? Interstitial fluid, right? So it has to be false. You know? Okay. ECF contains ECF contains these anions, right? Chloride and CHO3, right? So this is true. This is true, right? The CHO3, that's right. This is bicarb, right? Bicarb. And ECF contains one more and sodium also, right? That would be the cation. That's good. In control mechanism, which cycle is dangerous and may lead to death? Right now, in control mechanisms, we saw that there were two types: the positive feedback and the negative feedback. Correct? That's right. Positive feedback cycle. Right? So that is dangerous. And this positive feedback, when it leads to a cycle which is called as the vicious cycle, very true, vicious, vicious cycle. Vicious cycle stands for that when A increases, it leads to increase in B, and this B leads to increase in A, and it moves on and on and on and on, and finally it leads to death. Very good. Give an example of positive feedback mechanism useful for the physiological process, right? I think you must have picked this question correctly. The best example was that is during parturition, that is during the baby's birth, right? It is the uterine contraction, right? Uterine contraction. And that uterine contraction which leads to the pushing of the baby's head, and cervix stretches, and this stretch of cervix leads to further intensification of uterine contraction, and it moves on. Yeah, that's fine. Crowning, childbirth, birth of all good, all good, right? This I just explained, but all good. True. Next one was, who is responsible for the grain? Again, this is, तो अगर जो किसी ने गलत बोला तो remotely डंडा. Ribosomes. Correct. Yeah, this question is also like that. If anyone has answered this thing incorrectly, to gaya, right? So, correct. Which part of cell membrane is lipophilic? Right. It is this central part. Correct. Central part. This one is outer. This one is inner. So this central part, that is middle. Middle or central. Anything. Middle is also fine. Right. That one is lipophilic. It loves lipids. Sphingolipids, they are present in which type of cells? Sphingolipids. Right, sphingolipids, they are classically into nerve cells. But nerve cells. Right? Nerve cell. True. And the question number 9. Give any two functions of glycocalyx. Right? Those glycocalyx, which are like, which was like, it is negatively charged. It keeps the membrane negatively charged, right? Because of this, so those cells, they are attached with each other, right? Attached with each. Then this immune system, very right, right? It takes part into immune system, right? And one more was that it acts as a receptor, right? Anyone who has written this receptor, yes, what you wrote is very right. Receptor for binding insulin, fantastic. It acts as a receptor for binding insulin, right? Very good. See, you guys study so well. I'm very happy. Very nice. What is synthesized by RER? Rough endoplasmic reticulum. When he is rough, so we remembered it like that healthy tips, they are always rough. No one likes it, right? So that is the protein, right? That is the protein. Correct. And those smooth endoplasmic reticulum, right? They are all burger pizzas, right? Lipids, right? Lipids. And then only thing which is left out is the carbs. And the carbs are by. Oh, there is one question also, right? There is one question also on that. No problem, we answer it right now. So carbs are synthesized by someone who is synthesizing carbs is because carbs cannot be in Golgi body. That's right. Golgi body. Right. Correct. Hydrolysis of lipids leads to formation of which substances? Right. This was good because proteins, they lead to amino acids. Correct. Amino acids. So far it was okay. Then glycogen, that would lead to glucose. Correct. But we are asked lipids. So for lipids, yes, fatty acids and glycerol. Very right. Right. It leads to fatty acids plus glycerol. Good one. Good one. Huh. What is a function of peroxisome? Right. The peroxisome, right? Uh, you wrote that Gunda cousin, right? Because of that hydrogen peroxide, what it really does is, yeah, detoxification, right? It uses that process of it uses oxidase, right? And thus it leads to detoxification, right? Detoxification. For this detoxification, it would use what? Removes poison by one enzyme, and it is catalase, right? It's fine if you have written this. If not, no worries. Catalase, right? And that's how it detoxifies. Yes, it self-replicates. True, it's self-replicating. Right? That is the characteristic. We can write it over here. Self-replicating. Very true. Self-replicating. Right? And then that's how it converts this alcohol to acetaldehyde. You know? Acetaldehyde. But not fully, right? It's almost the 50%, right? 50% of alcohol that is done. Yes, 50% alcohol. This though it was ekdam easy. You know? Christe, Christe, right? Again, this is like a danda question. किसी ने गलत किया तो, right? In a cell, mitochondria are fixed in numbers, right? Ha ha ha. You know? If that is the case, never do the exercise, right? We discussed so much about it. Yes, it is false. Because with exercise and as the demand increases, not only the number, but also the size, right? So there is increase in number and there is increase in size. Both increase, right? So this is false. Good one. Next is, yeah, what is Desmin? Right? What is Desmin? What have you written in this answer? Answer of this question number 15. What is Desmin? Mechanical support in muscle cell. Very nice. Very nice. In fact, that is a very, very good, right? That's right. This is basically a support filament. Very good. Very good. Support filament, right? Filament in muscle cell. Good. Nice. Intermediate filament, that's right. That is also true. Fibular protein, that's also true. Okay, name the substance forming boundary of the nucleolus. Which substance? Which substance forming boundary of the nucleolus? Hmm. Jaidev, nuclear membrane. Shabash. This was googly. Acha. Good, Sally. Good, Siddhant. Okay, no limiting boundary, no boundary. Because it was not nucleus, it was nucleolus. Nucleolus has got no boundary, right? Because it is just a clump of, RNA. You know? So it has got no limiting boundary. Right? Ha. Give three characteristics of pinocytosis. Pinocytosis, pin, small one, right? So one was that it's very fast, very rapid, right? Then it keeps on happening, right? Continuous, very true. It is continuous. Then it is only for those small particles, right? It is for very small particles. True. Very rapid, very small, easy to go. True, easy to go. And that's how the proteins, they enter into the cell. Easy to go. Good. What is the function of lysopharin in lysosome? Right? What is the function of lysopharin? The answer, the hint. Yes, very nice, very nice. Pharine, right? It is to dissolve the cell wall. No, 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 no. Not to dissolve the cell wall, because that is for lysozyme. This is lysopharine. Yes, because it binds. It binds iron, right? Ferrous. It binds iron plus other substances which are needed for bacterial growth. So when it binds iron, those things, they are not available for the bacterial growth. So what it really does is, this bacterial growth is stunted. It is stunted. So bacterial growth is stopped. Correct. By which processes? You are asking about the processes. SER, smooth endoplasmic reticulum enzyme, do the detoxification. Right. This is like a quite a lengthy. Yes, so oxidation is one. That's right. It is oxidation. Then coagulation. Yes, coagulation, oxidation, hydrolysis. Good. So it is coagulation, oxidation, and hydrolysis. Very good. Hydrolysis. That's right. What is synthesized by high? This we already talked about, right? Carbs. Let's move on to question number 21. Yes, fill in the blank. Right? Here. What was this X? This one. It is carbohydrate, lipid, proteins. Yeah, very good. Pyruvic acid. Good. It is pyruvic acid. That's right. Name the cations which are present in ICF. Cations which are present in ICF, right? So cations means these are all positive, right? So potassium and magnesium. Very good. Very good. ECF is double than ICF. True or false? ECF is double. Cl minus and HCO3 minus, right? See, these are all anions, right? A negative ion, right? A negative ion, right? That's why I asked this question. See, anions means we remember it like a negative ion, right? It means they are negative ions. Here, cations. So only the positives they were asked. Huh. ECF is double than ICF. Right? That rule, and when we say that total body water is 60%, right? And out of that 60%, most of them into intracellular fluid. So intracellular fluid is about 40%, right? And extracellular fluid is 20%. So in fact, it is other way around. ECF is not double, but it is half the ICF, right? That's right. So this will be obviously false. Name the substance used to estimate the total body water by dilution method. What was the answer? Right? The heavy water, right? It is like deuterio, which is D2O, also called as the heavy water. Heavy water, right? That is what is used. And even there are even other substances like tritium, etc. So if you have written about tritium, that's fine. If not, no problems. This D2O, that was the main, right? This is total body water, so it will be going when you inject it, right? It will be going into plasma. From plasma, it will go into interstitial fluid. From interstitial fluid, it can penetrate even into the intracellular fluid. Thus, it gives the total body water. Correct? Question number 25. Inulin, right? Inulin cannot cross the cell membrane while mannitol can cross the cell membrane. True or false? Okay. Anyone else? False. That's right. That's right. It is false. Because inulin to is okay. Inulin cannot cross the cell membrane. But mannitol also cannot cross the, right? Both can't. Right? So both cannot. Good. Good. What is Gibbs-Donen effect? Right? Were you able to explain this? Was this is how we really discussed it, right? So this is the thing. This is semi-permeable membrane. Here is bada sa, like say albumin who is sitting and he is negatively charged. So he'll be right. You know what came in my mind? He'll be marring dhakka to all those negative. But that's how we remember, right? So he'll be marring dhakka to negative. So that means it will be repelling, right? And it will be, say, all those positives, right? Those cations, they will be attracted, right? It would attract. And this process will continue till we get the electroneutrality, right? That is what is called as the electroneutrality. It continues till this point. So this is the reason that in both the compartments there won't be exactly the same ions, right? But there would be some variation. This variation would not be as big as compared to ECF and ICF, right? But there would be a minor difference between the plasma and the interstitial fluid, right? This whole thing is called as the Gibbs-Donen effect. This was like a language question. What do you mean by ubiquitous? Ubiquitous means something which is present everywhere. That's right. Present everywhere. Good one. Name the substance to estimate the intracellular fluid. Right? If anyone has answered this thing, right, we would like to give him or her the next Nobel Prize. Jaydev, fine. The Nobel Prize goes to you. I was about to say that, huh? Right. Anyone else? Any more candidates for Nobel Prize? Hmm. Huh, that's fine. But here is only intracellular fluid. बस और कुछ नहीं. Yeah, exactly. No substance, right? There is no substance. None. Answer is none. Because there is no substance which has ever been discovered which goes only into intracellular fluid. So imagine, yeah, you have injected it into blood. So then this entire substance, it goes into interstitial fluid, right? Interstitial fluid. Then this entire substance goes into those cells, right? All these cells, they are filled with this substance. How will we act, even if there is any substance, how will we measure? In fact, there is no substance which goes only into intracellular fluid, right? That's why the method to estimate intracellular fluid is indirect. You measure the total, you measure the ICF, you measure the total, you measure the ECF, and that's how you get the ICF, right? Because total is ECF plus ICF. Now, because this cannot be measured, so that's why you take it on this side. So total minus ECF, and that's how you get ICF. And this is what is called as the indirect method, right? Indirect method. Okay. Calculate the osmolarity of a solution with following details. This was pretty easy, but still, I would like to know your answers. What was the answer? 10. Siddhant, why 10? 10, 13, 8, 10. What is happening? How come? How come 10? 2 twos are 4 and then the 6. Huh? How come 10? 12? 12? ये कैसे? How 10, 12, 13? Sodium is 1, chloride is 1, this is Ca, this is Cl minus, this is Cl minus, and so this is 2, right? These are 2, so 2 into 2, so this one is 4. This one, 1, 2, 3, 3 into 2, so this one is 6. And glucose is just 1, right? 1 glucose, so glucose is just 1. है ना? 11, that is correct. That is correct. DK, you wrote 146. That's right. 146, just the total. That's right. Okay. Happens. Chalo. Fill the below details when hypotonic solution is added. Hypotonic solution is added. So any solution, it is added into extracellular fluid. So this will increase, correct? Okay. So this is, right, it will increase. When you are giving hypotonic solution, so amount of salts which are inside, right, they will actually relatively drop. So what will happen to osmolarity of ECF? What will happen to osmolarity of ECF? It will increase or decrease, because relatively the number of salts which are over here, they will be increase or decrease. They will increase or decrease. Osmolarity of ECF, right? Decrease. Very right. Very right. When the osmolarity of ECF decreases, that means there are relatively more salts into the ICF. So fluid will start transferring from this is, if this is ECF, this is ICF, so it will start transferring from ECF to ICF. So there is increase in the ICF. Now in ICF, more water is coming, the number of salts they are fixed, so obviously the osmolarity is going to drop, right? So that's how we get it typically into hypotonic solution, right? I hope everyone has answered this thing correctly. Good. On to the next one. This is when hypertonic solution is lost. Hypertonic solution means simply salts are lost, right? Salts are lost. More salts are lost, right? This is typically in those Addison's disease, right? Say Addison's disease, mineralocorticoid disease, Addison's disease. Okay, so first thing first, hypertonic solution is lost. That means fluid to gaya hai, it is decreased. So chalo, it is decreased. But because we are losing more salts also, so that means osmolarity should also drop, right? Because salts, more salts are lost. Fluid is lost and the more salts are lost, so osmolarity drops. Universal principle, it will start transferring. ICF will feel कि अच्छा there are more salts and I have got less, so let me transfer the fluid. So this fluid, right, this ICF will start transferring, right? It will increase, correct, right? And when the fluid is moving into ECF, right? Fluid is moving into ECF, so there will be relatively more percentage of this ICF salts in ICF, so that will also drop, right? Because more fluid, so it is less osmolarity. Good. This was easy. This was easy. Which are the three yes? Which are the three criteria or the three conceptual criteria responsible for the substance to cross the cell membrane, right? One was that if the substance is lipid soluble, so it would go easily, right? Second, we said that if it is having small, yes, size, the small molecular weight substances, right? So they can cross easily. And finally, the uncharged or the neutral. That's right, neutral, right? Neutral or uncharged particles, they will be going easily, right? The basis of so many drugs, whether they will be going, crossing or not. In which part of the cell membrane phosphatidylserine is normally present? What was the answer of this? What did you write in this? In which part of the cell membrane? That's the cell membrane, right? Lower part, yes, lower part or inner part. That's very good, right? It is because of the membrane asymmetry, right? Membrane asymmetry. This is only the inner part or the lower part. Anything is fine. Or the ICF. That's also granted, right? So inner, lower, ICF, all good. So this phosphatidylserine is present over here. Acha. By the way, what if this phosphatidylserine appears on the outer side? Then what? What would happen if it appears on the outer side? This is outer side. What would happen? Suicide. Yeah, actually it is like that, right? Because this cell will go into apoptosis, right? This cell will go into apoptosis and it will say, eat me, right? Eat me. It is the eat me signal, right? Right? Not eat me single, right? It is eat me signal. So that's that, and the cell goes for apoptosis, right? And it goes. That's right. Next one. Again, this was the cakewalk. Saturated fatty acid increase the fluidity of cell membrane, right? Saturated, they are not good, right? So they in fact decrease the fluidity. So this is false, correct? in membrane of which cell is the presence of more lipids as compared to proteins? I think you must have answered this also correctly, right? Because we had a discussion about it. Which cell the presence of, and this is like a question which is asked in competitive exams. In which type of cell? Myelinated nerve cells. Very nice answer. Precisely. This should be the answer. Yes. Because if you have written only nerve cells, so then there is bit of controversy. Myelinated, non-myelinated. It is myelinated. That's right. Right? It is myelinated nerve cells. Good one. Nerve cells. And right, in myelinated nerve cell, the proportion is extremely different. It is 80%, and over here it is just hardly 20%, right? So many lipids. So lipids, they are 80%, and over here these are 20% proteins, as compared to when it is in the ratio of about 50-40, 55-40, right? Normally. So normally it is about, say, 55 proteins and 45 lipids, right? But it is myelinated nerve cells. That's right. Exceptional case. Very true. This is the exceptional case. Name the major surfactant. Major surfactant, right? No one should miss this thing because it is phosphatidyl. That is also true. Phosphatidylcholine, which is also called as the lecithin. Lecithin. So anything if you have written phosphatidylcholine or lecithin, both the answers are right. Which ratio is vital to check the fetal lung maturity? Right? It is the LS ratio. Lecithin sphingomyelin. Right? Lecithin sphingomyelin. And what should be this ratio? It should be greater than or equal to 2. So that is the indication. Yes, more than 2, lecithin sphingomyelin. And this test is also called as, what is the name of the test? I am not asked, but still, what is the name of the test? Name of this test in which amniotic fluid is taken. Clement test. Very good. It is Clement test or shake test or bubble test. Fantastic. Right? It is shake test or bubble test or the Clement test. Clement test. Nice one. Nice one. Which antibody is detected in VDRL test? Name of the antibody which is detected in VDRL. It is, yeah, that's right, cardiolipin. Not clarithin, cardiolipin. Then define receptors. Well, receptors are all those macromolecules, right? These are the macromolecules, and these are all surface proteins, right? Surface proteins on which the ligand acts, and it's in such a way so that they send signals inside the cell for the biological action, right? Biological action. And these ligands, they could be some drugs, some neurotransmitters, they could be some enzymes, right? Toxins, chemicals, right? All of these, they will act, right? They pass a signal, commands, yeah, hormones. That's right, I missed that. Hormones, true. So they lead to that biological action. Yeah, they can act as a recognition site. The next one is GPCR. Receptors are also known by 2 other names. Those 2 names are, this was easy, right? 7 pass receptors, or also called as that's right, serpentine receptors. Serpentine receptors. That's right. Question number 44. What is this phylog protein, right? So this is guanosine, right? That was our phylog protein. I think no one must have missed it. What is the effect of inhibition of adenyl cyclase enzyme, right? Adenyl cyclase enzyme, the final one, right? So adenyl cyclase, it converts that ATP into cyclic AMP, because it is cyclic AMP, which is like a functional currency, right? Which is like a functional currency. So when that's the function of adenyl cyclase. So obviously when it is inhibited, so this function is inhibited, right? Inhibits ATP to cyclic AMP conversion. Very nice, very nice. Name of the enzyme inhibited by drinking tea or coffee. What is name of that enzyme which inhibits tea or coffee? Name of the enzyme is phospho, because the reverse of that, right? Phosphodiesterase. Diesterase. Because this phosphodiesterase, what it does is that this CAMP, right? Cyclic AMP is active, right? This AMP, right? So it really stops over here, right? It really inhibits. When say this enzyme, right? This enzyme would be inhibited because of tea or coffee. So this remains active and it keeps things good. Active. Phosphodiesterase. That's right. The next one is protein kinase A activates the enzyme to stimulate basic body metabolism by which method? Was by giving this, right? This is what? Phosphorylation. That's right. Phosphorylation. Everything, whether it is on the genes, right, or it is onto the enzyme, basic function, phosphorylation, and that's how it really works. What do you mean by mydriasis? Well, mydriasis is when those at contraction towards the surface, right? Contraction towards the surface. So it leads to dilated pupil. That's right, right? Dilated pupil. Hmm. Where do you find beta 2 adrenergic receptors? Lungs, right? That's right. Lungs. Perfect. Name the 3 factors affecting diffusion. That's true. That's true. Where do you find that's true? Where name 3 factors affecting diffusion. Which 3 factors which are affecting diffusion? Right? First one was concentration gradient. Correct? Yes. So concentration gradient. Wherever it will travel from high concentration to lower concentration. Then second is membrane, right? Yes, size and pressure, membrane surface, right? Membrane surface. So if the surface is more, right, there would be more transfer, membrane surface area. And finally, that inversely proportional to thickness, right? Thickness of membrane. Okay. List the location of GLUT type 2, right? This is type 2. So type 2 is alpha is 1, beta 2. So beta cells, right? Beta cells of islets of Langerhans, and that is pancreas, correct? Then yes, two kidneys, right? So that's how we remembered, two kidneys. Then liver, because liver has got two lobes, right and left, so two kidneys. And finally the intestines, right? Intestines, excellent, right? Because we drew the intestines like something like this, right? And then like this. So it is present in intestines. Good. Which syndrome results in mutation of GLUT2? Syndrome which results in mutation of GLUT2. That's right, Fanconi, Bickel, and these are the name of the scientist, right? These are the name of the scientist. Bickel syndrome. Syndrome, right? Also, like, it is a sort of glycogen storage disease, right? So just to know, right? It is glycogen storage disease. List the location of GLUT type 5. GLUT type 5. So we wrote it like seminiferous tubules in testis. And very good, GIT, right? Seminiferous tubules and GIT. Okay. Is this correct? Right? We have shown sodium potassium pump. ATPase is needed. Yes, it is needed. ATP is needed. Yes, it is needed. Cell membrane is there. Yes, it is there. Sodium to potassium, right? Sodium to potassium ratio. This is 3:2. Very right. So is this correct? Yes or no? Yes. Okay. Yes. Okay. That's right. This is not correct because the direction has been changed. Exactly. Direction is opposite. It is the sodium which is going out because outside there is so much of sodium, right? And ATP is needed because you are going against the pressure gradient, right? So sodium goes out and it is the potassium which comes in. So they all are working against the concentration gradient. So that's why this is not correct. Good. Which part of nephrons is responsible for the reabsorption of filtered glucose back in blood? Very straightforward answer. And this is efflux, PCT, right? PCT, proximal convoluted tubule, right? So this is like our nephron, Bowman's capsule, right? And this is the proximal convoluted tubule, and then it goes into the form of loop of Henle, and then you have got the distal convoluted tubule and the collecting duct. It is this portion, proximal convoluted tubule, where the glucose is filtered, and over here it is reabsorbed back, because if this portion is missed, so then glucose gaya seedha blood mein, right? So this is the PCT, proximal convoluted tubule is the answer. Good one. Your diabetic patient is on canagliflozin. So which common complication you will watch for? Which common complication will watch for? Heart rate will fall. Okay. UTI. Okay. Presence of glucose in urine. Right. UTI. That's right. See, because canagliflozin will block actually this PCT. So it won't allow PCT to take that glucose back in blood. So it will keep on going into the urine. So that's fine. Glucose will go into the urine, but the complication is because glucose is the best culture media, insulin level will drop, surely. But the idea is because the glucose is pushed into the urine. Now, when it is pushed into the urine, you are not telling, you are restricting that reabsorption of glucose back into the blood. So that's why in blood the glucose level would fall. That's what you really want in diabetic patient. But because it is going in urine, and this glucose is a very good culture media, right? So this glucose, as it goes into the urine, and why it is because at PCT you have stopped reabsorption, right? So that reabsorption of glucose that has been restricted, it goes into the urine, and this is what leads to urinary tract infection, right? Very, very important, right? Glycosuria, fine. This glucose is going in urine, but it will lead to this problem, right? Urinary tract infection. So that is the commonest complication which we would really see. Why cardiac arrhythmias can occur in Gentleman syndrome? Because this thing will lead to increase in serum, which iron? Which iron? Excellent. This would be increased serum calcium, or the calcium in blood. And when the calcium is more in serum, it will lead to abnormal cardiac activity. Right? So that's the reason. And finally, this indicate increase or decrease of ions in Bartter syndrome. Yes, calcium, that's right. So indicate increase or decrease. So let's start with the serum one, right? So in serum, calcium will increase or decrease. That's right. It would decrease. This is the reason that in Bartter syndrome, we won't be getting cardiac complications, right? Arrhythmias. What about the urine? Yes, it would increase. That's right. That's right. And what about the magnesium? Yes, a magnesium can increase or it can be normal, right? It can increase or it can be normal. That's right. So this was our last question, right? Done. So, how was your performance? Out of 55, we just consider 1 mark for every question so that it becomes easy. Okay, 52. That is very nice. Okay, 43. 57, 51. Good, good. 51. Okay. 48, not bad. 49, 45, 49. So quite nice. Good, good, good one. So from Monday, we'll start further topics, right? Wish you a very happy weekend. Thank you so much. We will not discuss that which country will win the FIFA World Cup, right? So take care, and we meet on Monday, same time. Thank you and bye bye. Enjoy the weekend. Next topic, we'll be starting with the action potential. And next week, we'll also take one session on अपना receptors, right? Okay, thank you and bye bye.